Hello everyone and welcome to our module on amino acids. Amino acids are the building blocks of proteins. Proteins are polymers made up of monomers of amino acids, and every amino acid contains two important groups. One is called an amine group and one is called a carboxylic acid. If you look at the structure on the left side of the screen, this is the generic structure of an amino acid. So there is a carbon in the middle, which is called the alpha carbon. Attached to the alpha carbon is a nitrogen group and several hydrogens. That's the amine group. Also attached to the alpha carbon is a COOH group. That's the carboxylic acid group. What makes one amino acid different from another is this R group here that's attached to the alpha carbon. Every amino acid has a unique group that is attached to that alpha carbon. In the case of glycine, which is the simplest amino acid there is, what's attached to the carbon is just a hydrogen. So there's just a CH2 molecule here attached to the amine group and the carboxylic acid. Other amino acids have much more complicated R groups attached to that alpha carbon. Shown on this slide are the 21 amino acids that are encoded by our genome, and you can see that they have very different side groups. This is glycine right here, and it's very simple like what I showed you on the last slide, but other molecules have much bigger and more complex side chains, like tryptophan here has 2 rings. Histidine has a ring as well. Some of them have a long side chain like lysine. But every one is the same in that it has a COOH group, a carboxylic acid group, and an amino group, an NH2 group, attached to an alpha carbon to start the molecule off. It is those side chains that make one amino acid different from another. If you took organic chemistry in college, you probably struggled to learn about enantiomers. Those are molecules that have the same molecular makeup but are mirror images of one another. Luckily, you don't have to know a lot about that for medical school, but you do have to know that all amino acids except glycine have L and D configurations. On the screen here, I've drawn L-alanine and D-alanine. They are mirror images of one another. These are called enantiomers. And you should know that only the L amino acids are used in human proteins. The D configurations are never used. And glycine is the only amino acid that does not have L and D configurations because its structure is so simple that you can't make a form of it that is a mirror image of another form. That is not true for all the other amino acids. For the next few slides, I'm going to talk about the acid-base behavior of amino acids, and we're going to start by defining the term pKa, which is the log acid dissociation constant. So for any acid, which I've noted by the letter A here, that acid can exist in solution in 2 forms. In one form, it can be holding onto a proton. That's called the HA form. In the other form, the acid can dissociate from the proton and exist in the A negative form and release the proton. And this is how acids, including amino acids, behave in solution. Sometimes they are found in the HA form, and other times they are found in the A negative form with the proton having been released. So when amino acids and other acids behave like this, we can define a term called the pKa. And we can say that the pH is equal to the pKa, which is a dissociation constant that's unique to each acid, plus the log of the concentration of the A negative form divided by the concentration of the HA form. You can also rearrange this as I've shown down below so that pKa is equal to pH minus the log of this term. Well, anytime in solution you have a concentration of A negative that is equal to HA, then this term is going to be 1 and the log of 1 is 0, so this term won't matter. So we can measure the pKa for any acid, including an amino acid, by creating a solution where the concentration of HA and A negative are equal, and the pH at that point will be equal to the pKa. So this is how we define pKa values experimentally for any acid, including amino acids. For step 1, they're not going to ask you to calculate pKa values, but they are going to want you to understand how an amino acid will behave when it's in a solution where the pH is higher or lower than the pKa value. So in order to understand that, remember that an acid can be present in solution either in the protonated form, HA, or the unprotonated form, A negative, and which form is favored will depend on the pH of the solution. So when the pH is higher than the pKa, you can think of this as a solution where there are relatively few hydrogen ions around. A high pH means relatively few hydrogen ions around. And when there are few hydrogen ions around, you can imagine that there are going to be very few molecules to bind to the acid. Therefore, you're going to predominantly find the acid in this form, the unprotonated form. There just aren't many H pluses around to go and bind to the acid, and therefore this is the dominant form when the pH is higher than the pKa. On the other hand, when the pH is lower than the pKa, you can think of that as a solution where there are lots of hydrogen ions around. Remember that a low pH means lots of H plus molecules in solution. Therefore, there are lots of molecules around to bind to the acid, and therefore this will be the dominant form. Even though both will be present, you will find it more likely in this form when you are in a solution where the pH is low relative to the pKa. So to make this point clear, let's consider a very simple acid that is not an amino acid. Let's talk about acetic acid, which has a pKa of 4.75. This is the structure of acetic acid on the screen here. So if the pH in solution is lower than 4.75, is lower than the pKa of acetic acid, this means that there will be lots of hydrogen ions around. A pH less than 4.75 is very low. There are going to be lots of protons floating around in solution. So this is going to be the form you're mostly going to find at pHs less than 4.75. On the other hand, if the pH is above 4.75, this means you have relatively few hydrogen ions around, as I've shown in this picture here. So therefore, this is going to be the form of the acid you will be more likely to encounter. Let's also consider the case of ammonia, which has a very high pKa of 9.4. So if the pH is less than 9.4, you can just think of that as a solution that has lots of hydrogen ions around relative to where ammonia likes to be in equilibrium. This means you're going to be more likely to find the NH4 form where the ammonia is protonated and has a positive charge. On the other hand, if the pH is greater than 9.4, you can just think of this as having relatively few hydrogen ions around compared to where ammonia likes to live in equilibrium. Therefore, you're going to be more likely to find the NH3 unprotonated form of ammonia. In contrast to acetic acid and ammonia, amino acids are complicated because they have multiple acid-base regions of the molecule, and each has its own pKa value. Every amino acid has a carboxylic acid group, which is a COOH group, and it can dissociate into a COO negative group plus a proton. The carboxylic acid groups tend to behave like acetic acid. They tend to have low pKa's, usually under 4. So at normal pH inside the plasma of 7.4, you usually find them as COO negative molecules. The amine groups, on the other hand, have an R attached to an NH3 plus group, which can dissociate into an NH2 group plus a hydrogen atom. These usually have a high pKa greater than 9 or so, and so at normal plasma pH of 7.4, you will be more likely to find them in the NH3 plus form. To make amino acids even more complicated, sometimes the side chain has a pKa. This means the amino acid will have 3 different pKa values. Consider the case of lysine. It has a carboxylic acid group, like all amino acids, with a low pKa of 2.18. It has an amine group, like all amino acids, with a pKa of 8.95, but then it also has a side chain which ends in an NH3 group, and that has a pKa of 10.53. You can determine the pKa's of the different portions of an amino acid by performing a titration experiment. In a titration experiment, you add increasing amounts of sodium hydroxide, which is a base. In solution, the sodium hydroxide will form molecules that are OH negative. They will tend to pull hydrogen ions out of solution to form water. What this means is, as they pull hydrogen molecules out of solution, the pH will start to rise, because anytime you have fewer hydrogen molecules in solution, the pH begins to rise. So as you can see in this curve I've shown on the left side of the screen here, as you add sodium hydroxide, the pH starts to rise, like in this portion right here. However, when you reach the pKa of one of the parts of the amino acid, the curve starts to flatten out. Why does that happen? This happens because part of the amino acid molecule is acting as a buffer. In the case of the low pKa here, what's happening is each time the OH negative group pulls an acid molecule out of solution, that acid molecule is given back to solution by the carboxylic acid group. That's why it's acting as a buffer, and the pH isn't changing until you give enough sodium hydroxide to pull every hydrogen molecule off of every carboxylic acid group. Then the pH will begin to rise again, and then it flattens out again when you reach the second pKa of the amino group. In molecules that have 3 pKa's, you would see a curve that looks something like this, and each flat portion represents the pKa of one of the portions of the amino acid molecule. If we know the pKa value of the different portions of an amino acid molecule, then we can determine what charge that molecule is likely to have at normal plasma pH. So remember that normal plasma pH is 7.4, and let's start off by considering the case of glycine, the simplest amino acid. Glycine has a carboxylic acid group with a pKa of 2.2 and an amino group with a pKa of 9.6. So plasma pH is in the middle. This means that the carboxylic acid group is likely to be deprotonated and have a negative charge, and the amino group is likely to be protonated and have a positive charge. And the negative and positive charges will cancel out so that overall glycine at plasma pH usually has no net charge. Contrast that with arginine, which is an amino acid with 3 pKa values. The carboxylic acid group has a low pKa of 2.01, so at plasma pH you are most likely to find this unprotonated with a negatively charged oxygen molecule. The amine group attached to the alpha carbon has a pKa of 9.04, so at plasma pH this is most likely to be protonated with a positive charge, and the positive charge from this amine group will cancel out the negative charge from the carboxylic acid group. However, the side chain also has an amine group with a very high pKa of 12.48. At plasma pH, you are most likely to find this protonated, which means it will have a positive charge. This means that arginine at plasma pH has a net charge of +1, a negative charge for the oxygen molecule here, but a positive charge for this amine group and another positive charge for this amine group, giving it a net overall charge of +1. This also means that at plasma pH, arginine will remove one hydrogen ion from solution. Let's talk about why that's the case. This oxygen group will donate a hydrogen ion to solution. However, this amine group will take one out, and these two will cancel each other out. Then the side chain will take an additional hydrogen molecule out of solution at plasma pH. This means that on balance, there will be a net removal of one hydrogen ion from solution by the amino acid arginine. And anything that removes hydrogen ions from solution is called a base, and therefore arginine is a basic amino acid. There are two basic amino acids, arginine and lysine. Arginine is the most basic. It is the strongest base because its side chain has a very high pKa. Lysine is also basic with a side chain that has a pKa of 10.53. Both of these have a +1 charge at normal pH, which means they tend to remove one H plus from solution at normal pH. And when you remove H plus from solution, you raise the pH, and that makes these basic amino acids. You can find large quantities of basic amino acids inside of histones in the nucleus of cells. I discuss histones in more detail in the module on DNA structure, but for our purposes here, you should know that histones contain basic amino acids. They have a high content of lysine and arginine, which, as we just discussed, are positively charged amino acids. This makes them very favorable for binding the negatively charged phosphate backbone of DNA. This is the structure of adenosine monophosphate on the screen here. It's one of the nucleotide building blocks of DNA. And just like all nucleotides, it has a phosphate moiety attached, which has a negative charge. This can easily bind to the positively charged amino acids of lysine and arginine, and this makes these amino acids very useful when the DNA is trying to wrap around histones, as I've shown in this picture here. Histidine is another amino acid that's considered a basic amino acid. That's because its side chain can accept a proton. However, the side chain has an unusual pKa of 6.0 that's very close to plasma pH of 7.4. So you tend to find this unprotonated in solution. This means that usually at plasma pH, histidine will have no net charge. The negative charge here will be canceled by the positive charge here, and the side chain will not be carrying a proton. Now that we understand basic amino acids, let's discuss acidic amino acids. There are two acidic amino acids, aspartate and glutamate. Let's talk about aspartate first. So at a plasma pH of 7.4, we will be above the pKa of the carboxylic acid group here. That means we will be most likely to find this negatively charged. We will be below the pKa of the amino group here, so we will be most likely to find this positively charged. And this negative charge and positive charge will cancel each other out. However, the side chain also has a carboxylic acid group with a low pKa of 3.18. At normal pH, we will be above this pKa, so we will be most likely to find this negatively charged. This means that overall, the molecule will have a -1 charge. This also means that overall, this molecule will donate one hydrogen atom to solution, and anything that donates hydrogen atoms to solutions is an acid that tends to lower the pH. And both aspartate and glutamate behave in this manner at plasma pH. A number of amino acids have hydrophobic side chains. I've written the names of those on the screen here and also shown their chemical structures. It's very low yield to memorize all of these, but you should recognize that when large proteins are found in the plasma, these types of amino acids will be at the center, away from the plasma and the water that they don't like, and the hydrophilic amino acids will be on the outside coming into contact with water. You also frequently find these types of amino acids in the lipid bilayer of cell membranes. One clinical condition where the hydrophobic nature of amino acids is very relevant is in sickle cell anemia. As you may know, this disorder results from the substitution of the amino acid glutamate for valine, and glutamate is a polar amino acid and valine is a nonpolar amino acid. So you can just imagine all the problems that can occur in the hemoglobin protein when an amino acid that is expected to be polar suddenly becomes nonpolar. This can change the conformation of the protein, and that's what results in many of the symptoms of the disease. Another amino acid that's somewhat high yield to know about is proline. It has a 5-membered ring for a side chain with an amino group in it. This gives it a very rigid structure, and for that reason you find lots of proline in collagen, which provides support to tissues. There are 9 amino acids that must be supplied by the diet. The body cannot synthesize them de novo, and these are called essential amino acids. I think it's relatively low yield to memorize the names of all these, but you should be aware that some amino acids must come from the diet and others can be synthesized by the body. The final topic I'll discuss in this module is the concept of glucogenic versus ketogenic amino acids, and this has to do with the metabolism of amino acids when they're used as fuel sources. Glucogenic amino acids can be converted to pyruvate or TCA cycle intermediates, and this means that they can be converted into molecules of glucose via the process of gluconeogenesis. Ketogenic amino acids, on the other hand, are converted to ketone bodies or acetyl-CoA in their metabolism. This means that they cannot become glucose via gluconeogenesis. Now, most amino acids are either glucogenic or glucogenic and ketogenic. There are only 2 that are just ketogenic. We'll talk about those in a minute. To make this point a little bit more clear, let's review how glucose is synthesized via the pathway of gluconeogenesis. Recall that oxaloacetate, which is a TCA cycle intermediate, can be converted to glucose via gluconeogenesis. So therefore, any amino acid that can be metabolized to oxaloacetate can also be metabolized to glucose and is therefore a glucogenic amino acid. Also via gluconeogenesis, pyruvate is diverted to oxaloacetate, so any amino acid that can become pyruvate can further become glucose via gluconeogenesis. Oxaloacetate is part of the TCA cycle, so any amino acid that can be converted into a TCA cycle intermediate can then be further converted into oxaloacetate and glucose. Acetyl-CoA, on the other hand, can never become glucose. Why is that the case? Well, that's because acetyl-CoA donates 2 carbons to the TCA cycle, but those 2 carbons are removed in the form of carbon dioxide. So acetyl-CoA can never increase the net amount of oxaloacetate in the TCA cycle and therefore can never be converted into molecules of glucose. So any amino acid that is metabolized to acetyl-CoA cannot be metabolized into glucose. It can only be used to synthesize ketone bodies from acetyl-CoA, and therefore it would be called a ketogenic amino acid. And now many amino acids can be converted to acetyl-CoA plus another molecule that can go to glucose, and therefore they are referred to as both glucogenic and ketogenic. Some are only glucogenic and only a few are just ketogenic. The 2 amino acids which are purely ketogenic are leucine and lysine. They are both metabolized to acetoacetate, which is a ketone body, and acetyl-CoA, which, as I just explained to you, cannot be converted into glucose. Therefore, these are referred to as purely ketogenic amino acids, and they are the only 2 that are purely ketogenic, and they are both essential. And that concludes our module on amino acids.